Welcome everybody. So in our last lesson we looked at gene expression and how a DNA code can be transformed into a protein product. And today we're going to be looking at gene regulation, so chapter 2, C and E, and how that process of transcription and translation can actually be regulated. So we've learned about DNA, proteins, the relationship between those, and how the protein can be made to do its job outside of the cell. But this process isn't happening lightly, and it's really tightly regulated and controlled because all cells and all organisms, they want to conserve energy. So they don't want to be expressing things all of the time because it's quite taxing to do so. So it's really regulated and really controlled. So you need to think about how many genes are actually in our body. So if all of those genes were being expressed at the same time, all the time, it would be a disaster and probably would be just explode with too many proteins. So how does our body know when we need a protein? How do we stop and start this protein production? And why do we need to regulate in this way? So the first dot point for this topic is learning about the structure of genes. So we sort of touched on this when we were learning about transcription and translation because we needed to understand it. But hopefully going through exons, introns, and promoters in more detail here will help you to understand the last video. So we're going to be looking at gene structure in eukaryotes first. We'll be looking at both eukaryotes and prokaryotes here, which will be important for our next chapter. But right now we're going to look at eukaryotes, so animals, plants, multicellular organisms. So while different genes have different DNA triplets, they all share the same structure. So even though obviously the code is very different, they all share the same structure and they all have these common elements. So the first common element, which we looked at a little bit in our last video, was this promoter region. So you'll see it highlighted here in blue. So just sorry, FYI, this diagram and this diagram is the same sort of thing. The first one's just representing a little bit more detail and before RNA processing has occurred. So the promoter region is a binding site for numerous things, but in our last lesson we learned that it's the binding site for RNA polymerase. It can also be the binding site for DNA polymerase and other enzymes that are needed and responsible for various processes. So you'll see up in this top diagram here that it's sometimes called a tartar box. This is because the promoter is normally made up of a bunch of thymine and adenine bases, which is T and A, which kind of make this tartar code here. So it's sometimes called the tartar box, but it is the promoter. It's normally at the start of a gene sequence, and it's responsible for binding of those various things. We then, of course, have our introns and exons, which we learned about in our last lesson, but in a little bit more detail. We've got our exons here and we've got our introns here. So remember that exons are the coding regions. So these are the regions that are going to be transcribed into a protein. And then we have our introns, which are not needed for protein production. They sometimes are used for, I don't know, structural support, all those sorts of things, but they're not needed. So these are removed and these are kept in the final mRNA molecule. And you'll see down here that this is obviously after RNA processing because we only have our exons remaining. So exons are coding regions, introns are non-coding regions for our protein. We then have our termination sequence, which we learnt about when we were learning about transcription. So when this termination sequence is reached, that's when transcription ends. So this is the termination sequence here. So of course, as the name suggests, is at the end of our gene structure. And then sometimes we will have a operator. So the operator is not in this first diagram up here, but it is in this second diagram. It's this peach section here. So what an operator is, it acts as a binding site for various enzymes that are involved in gene regulation. So we'll go over that in a little bit more detail, but you can see that the promoter's there. That's where, for instance, RNA polymerase will bind. And then we have our operator. So a bunch of things can bind to this operator to either stop transcription from happening or start it happening, etc., etc. So that's what our operator's for. It's for that regulation part. So genes aren't active all the time, as we talked about, okay? And we see this in our everyday lives. So we do have some genes that are active all the time. So things that are involved in production of enzymes that help us break down food, or enzymes involved in cellular respiration for energy. But then there's other genes that aren't active all the time. So for instance, there might be some particular hormones that are transcribed and translated only during puberty, and then they're switched off all the time. So only certain genes are active within cell types as well. So for instance, your skin cells, they don't necessarily need the same proteins as nerve cells. So skin cells might make more proteins like keratin and collagen and things for structural support, whereas your nervous system cells would be making more neurotransmitters and things like that. So even though all cells have all the DNA, only some genes will be transcribed based off of their function. And the main purpose of this gene regulation, as we said, is to conserve energy. So the whole purpose is we obviously want to survive. The less energy we can expend, the more likely we are to survive. So having these genes turned off when we don't need them is going to help with us conserving energy. So there are two main types of gene that we're going to need to know a little bit about. We've got our structural genes and we've got our regulatory genes. So our structural genes are the genes that you probably normally think of, okay? So they're the genes that code for proteins that help with our structure or function of the cell. So enzymes and transport proteins and hormones and all of those sorts of things. So these are your usual run-of-the-mill genes that you probably think of when you think of genes, okay? So that's your structural genes. Then you have your regulatory genes. So the purpose of a regulatory gene, its whole purpose is to regulate the structural genes. Okay, so often you'll see that a regulatory gene might be upstream from a structural gene, and this regulatory gene will be turned on or off, which will in turn determine whether this is transcribed or not. So regulatory genes are responsible for the production of regulatory proteins. So we have things like repressor proteins, so they repress transcription from occurring. We have activator proteins, so they activate transcription to occur. And basically this is how we can turn gene expression on and off through these regulatory genes, and it will determine whether we need those structural genes or not. We also have gene structure in prokaryotes, okay? So you'll see that this looks a little bit different. So while eukaryotes have a bunch of structural genes that might be scattered all around, a chromosome or whatever, prokaryotes are a lot more efficient because they are smaller. They need to be even more energy efficient than we do. So in prokaryotes, the difference is you'll see multiple structural genes that will be arranged together. So for instance, if you have a structural gene and all of these genes are involved in respiration, let's say that, okay? In eukaryotes, these genes might be in all different sorts of places, but prokaryotes, being very efficient, they will block all of these genes that have the similar function together in something called an operon. And then these operons will be controlled by a common promoter and operator region, and also a common regulatory gene as well. So things that have similar functions will be lumped together into these operons. And this is very specific to prokaryotes. So now that we know a little bit about the gene structure of eukaryotes and prokaryotes, we're now going to look at a specific example of gene regulation in a prokaryote. So it's still really important that you know the gene structure of eukaryotes, but we're going to be focusing on prokaryotes for this specific example. So we're going to be looking at something called the trip operon, which is said here to be a simplified example of the regulatory process. Technically it is, but it's still quite complicated to understand, so I'll try my best to go through it slowly. So this is the example that Vika wants you to focus on when we're looking at gene regulation. Obviously gene regulation happens for many genes, but we're going to focus on this gene specifically. So the trip operon is it's an operon within prokaryotes, okay? And these genes are all clumped together in this operon because they all serve a common purpose, because that's what operons are. So all of these genes are involved in the production of the amino acid tryptophan. So that's why it's called the trip operon, because tryptophan for short is trip. Okay, so trip just means an amino acid, which we've learned about and we know what they are. So this only occurs in prokaryotes and bacteria. And all of these structural genes that share a common purpose are controlled by this common promoter, common operator, and of course this regulatory gene up here. So the entire operon is controlled by that regulatory gene, like I said, to conserve energy, and that's our main goal. So that's what the trip operon is. It's a series of genes lumped together that are responsible for the production of the amino acid tryptophan, and they are all regulated through the same regulatory gene. Now, this slide is a lot of detail. What it is I've just stretched this gene out to give you some more detail, especially in this what we call the leader region of the gene. Okay, so this is the regulatory gene, this red box here. Okay, and this is our structural gene. So these are our, the actual genes that code for tryptophan. Okay, and this is our regulator. So this is how we're going to regulate that whole process. So all I've done is I've stretched out that regulatory gene so you can see a little bit more detail. And this detail is going to be quite important to understanding how we actually regulate this gene. So we've got our promoter, that's where our RNA polymerase is going to bind for both our regulatory gene and also our structural genes as well. And then we've got a promoter, an operator, which again, same thing for each case. That's where our regulators and our repressors and our activators and enzymes and all of those things are able to bind. In our structural genes, so this is our trip operon genes. I've just put it as one square there, but this is our trip A, B, C, D, E, etc. So there's a bunch of genes in here that have all been clumped together for their same purpose. What we're going to focus on is this leader region in the repressor, the regulatory gene here. So there's a couple of things that I want you to take note of in this regulatory region, because this regulatory region is what's going to be transcribed and translated first before we can even get to these structural genes. So a lot of the action happens here for the regulation of the production of tryptophan. So I just want to draw your attention to these three regions here. So we've got region 1, and in region 1 we've got two codons that code for tryptophan. Okay, so remember that we have that codon table that we looked at, and there was several different codons that code for tryptophan. In this particular region, we have two codons that code for tryptophan. So in this region, when we are transcribing and then translating it, we're going to need to attach two tryptophan amino acids here. Okay, so make note of that. In region 2, well, actually between region 1 and 2, we have a stop codon. Okay, so remember that stop codon is what is going to stop transcription and translation from taking place. So this is an important point because we can't get past this point with this stop codon here, which is going to come into play a little bit later on. And then we have this attenuation region between region 3 and 4. So all this means is, well, all it is a bunch of A and U, so adenines and uracil pairings. So the key thing to take away from that is that adenine uracil pairings are very weak. So do you remember that the purines and the pyridamines, they link together, okay? But there was one, so we had the GC combo, which had 3 hydrogen bonds, and then we had the AU combo, which had 2 hydrogen bonds. So this is what makes this quite weak, or it's weaker than a GC bond. So we have lots of these weak pairings here, which is going to come into play a little bit later on. So I'm just spreading that out, showing you those 3 regions, and then when I refer to them, you sort of know where I'm referring to. Okay. So we have 3 different scenarios in which repression or lack of repression takes place. So the first scenario I want to draw your attention to is when tryptophan levels are high in the prokaryote. Okay, so that means that we have lots of tryptophan floating around. Because we have lots of tryptophan, we don't need to make any more tryptophan, okay? That would be a waste of energy to make more tryptophan. So what we're going to do in this scenario is we're going to repress, okay? We're going to stop or repress transcription from happening in the first place because we don't need it to happen because we've got enough tryptophan. So the regulatory gene for tryptophan that I showed you in the last slide, nice and spread out, is constantly expressed. So this regulatory gene is consistently always being expressed. The structural genes are not. So it's the regulatory gene that's always being expressed to produce repressor proteins. So that means that there's always these repressor proteins being expressed. The high levels of tryptophan that are floating around in the cell are able to bind to these repressor proteins. So you can see here, this regulatory gene, okay, it has made this green repressor protein here. When it binds to tryptophan, which is free floating in the cell, that causes a conformational change. So that just causes a shape change, okay? So when there's tryptophan present, it binds to the repressor and causes a change in its shape. This shape change allows the repressor to bind to the operator. So you can see up here, okay, that the repressor has bound to the operator region. Because it is bound to the operator region, we can see that our RNA polymerase here, this little enzyme here, we can see that it's bound to the promoter, but it is blocked, okay? Because that repressor protein is sitting there, RNA polymerase can't move any further down the gene. So therefore, transcription is prevented. So RNA polymerase is stuck and transcription of those TRP operon genes cannot happen. And that's good because we don't need it to happen. We have lots of tryptophan and we are conserving our energy. So just a little summary here. High level of tryptophan means there's no need for tryptophan transcription, which means repression, okay? So to wrap my head around it, I did a little diagram, and I strongly suggest you do the same because I know it's quite complicated to show this high tryptophan scenario. So in higher tryptophan scenario, we have repression. So that means no transcription is happening. So number one, all of these repressor proteins are consistently and constantly expressed by this regulatory gene. So here's all of our repressor proteins here. We have also high levels of tryptophan. Okay, so we know that's what's happening in the cell. There's high levels of tryptophan, which can bind to the repressor proteins. So here we're seeing the conformational change. Okay, our repressor proteins and our tryptophan are binding together, which causes a conformational change. Then, because this conformational change has occurred, this repressor protein has now the right shape to bind to the operator region. Okay, so see it's bound here. That means that RNA polymerase, which is this nice big enzyme here, is blocked and stuck at this promoter region. So that means that these operon genes cannot be transcribed and we conserve energy and no transcription occurs. All right, this next scenario is where it gets a little bit complicated, and that drawn out regulatory gene that I drew out for you starts to come into play. So repression completely stops transcription from even happening. Then we have this scenario. So this is when there's low levels of tryptophan in the cell, okay, or free floating. So that means that there's low tryptophan, not high tryptophan, low tryptophan, but tryptophan is still attached to tRNA molecules, okay? So why would it be attached to tRNA? Because we know that tryptophan is an amino acid, so tRNA is probably doing its job and collecting the tryptophan amino acids to add to a polypeptide chain somewhere else, okay? Translation of other proteins. So this is the gray area where tryptophan is low within the cell, but tryptophan is still very much present and attached to tRNA molecules. So the difference between repression and attenuation, which is what this process is called, is repression completely stops transcription from happening. So it doesn't even let it happen in the first place. While attenuation, it just stops transcription early on. Okay, it stops it early on. So it starts, but then it is stopped because we have tryptophan on tRNA. So we still don't really need that much tryptophan yet because we still have it on tRNA, okay? And we're really into conserving energy, so we don't want to make it until we absolutely need to. So we have our regulatory gene, which is here, okay? and we have that leader region amongst that. So remember we had region 1, 2, 3, 4, and 5 that we went through. So remember we had codons for tryptophan in region 1. We had a stop codon between region 1 and 2, and we had that weak region called the attenuator region between region 3 and 4. Okay, so that's what we've got in that region right now. So what happens is that RNA polymerase can start to transcribe because the repressor proteins are not binding to tryptophan, because remember tryptophan is low. There's low amounts of tryptophan inside the cell, so it's not binding to the tryptophan, it's not changing its shape, and it cannot bind to the operator. So that means that RNA polymerase here has free rein, and it can start to transcribe the leader region, okay? At the same time, this is something really cool about prokaryotes. We can't do it, but prokaryotes can. And this is really important for this concept, is that the ribosome is simultaneously translating. So as RNA polymerase transcribes, the ribosome is simultaneously translating that transcribed molecule straight away. So in eukaryotes, we have to do transcription, and then it travels to the ribosome, then translation occurs. This is all happening at the same time. Okay, that's an important thing to note. So ribosome and RNA polymerase doing its job at the same time. So RNA polymerase starts to transcribe the leader region, and at the same time the ribosome is translating into a polypeptide. Okay, tryptophan is present on the tRNA. So that means that the tRNA molecules can add tryptophan to the polypeptide chain. So remember that we have these trip codons in the region 1, and trip can be added. Okay, added, no worries, we've got it there. The ribosome is then therefore stopped at the stop codon. Okay, so remember that stop codon was there for a reason. It's to stop RNA polymerase at that region. Stopping at that particular point causes something called an attenuation loop, or a hairpin loop, or an attenuation structure to form. So this forms at that weak region. So that weak region, so here it is here, these little loop structures. This weak region of AU base pairings, because there's only 2 hydrogen bonds, so it's very weak. So these weak regions cause this DNA to pull into these attenuation loops, hairpin loops, whatever you want to call them, these structures. Okay, this pulling, this yanking of DNA together, causes RNA polymerase to basically not fly off, but it falls off the leader region. So this yanking makes RNA polymerase detach. So because RNA polymerase has detached, it therefore cannot transcribe the trip operon fully because it's fallen off from the yanking of these hairpin loops. Okay, and therefore transcription is stopped early. Okay, so here's a diagram that I have made. This will hopefully help you. So this is when there's tryptophan only found on tRNA molecules. So we call this attenuation. Okay, this is attenuation. So what happens is we have, step 1, we've got our RNA polymerase, which can start transcribing the leader sequence. Okay, this is because these repressor proteins that have been made, they have no tryptophan to bind to, so they don't change shape. Okay, which means they can't bind to the operator, and they can't stop RNA polymerase in its tracks. So RNA polymerase starts to transcribe the leader sequence. As we said, prokaryotes can do transcription translation simultaneously. So we have the ribosome simultaneously translating, as we know it does. So because tRNA has tryptophan attached to it, so you can see we've got little tryptophan amino acids that are attached to these tRNA molecules, they can be added to the chain. So here I've actually written out the amino acid sequence that comes about from this leader sequence. So we've got all these amino acids, and then we've got our 2 tryptophan amino acids. Because we have tryptophan on our tRNA, we can add them. So nothing happens, we add them as normal, and we continue on until we reach this stop codon. Okay, so because we stopped at this stop codon, so that means that RNA polymerase has stopped transcribing. And for reasons you don't really need to know, this stop codon, okay, it brings us to this attenuation region that forms these attenuation loops. Okay, so stopping at this stop codon means these weak AU pairings fold into these hairpin loops or these attenuation structures. Because they're folding, and when you fold something, the ends get pulled or yanked. Because they're folding, this pulling yanks the DNA apart, okay, which then therefore stops RNA polymerase from transcribing. So RNA polymerase gets to here, and then it gets yoinked off, flies away, okay? So it gets yoinked off at this attenuation region, which means that it cannot continue to transcribe the trip operon genes. Okay, so this pulling yanks RNA polymerase off and stops transcription early. Okay, so some transcription and translation has happened, but it hasn't been completed. So those trip operon genes do not get fully expressed because we still want to conserve that energy and we still don't want to make tryptophan just yet. Okay. We then have our last scenario, and this scenario doesn't have a name because it's just normal transcription translation. So this scenario is when tryptophan levels are at 0. So we have no tryptophan in the cell and we have no tryptophan on tRNA. So finally the prokaryote has to give in and it has to make some tryptophan to keep translating its proteins. Okay, so it finally has to actually do its job. So in this scenario, we want transcription and translation to occur. So when there is no more tryptophan or tRNA, the ribosome will stop at the trip codons instead of the stop codon. So remember we had our leader sequence and we had, all these amino acids, we had 2 trips, and then we had a stop up here. Because there is no tryptophan, so our little tRNAs have no tryptophan on them, we can't add these. So that means that the RNA polymerase is prematurely stopped here. Okay, so instead of stopping here, it stops over here. Okay, because it can't add those tryptophan, so it's stopped, it's halted, it doesn't know what to do. Because of the stopping in this particular region, something called an anti-termination loop forms instead of that attenuation loop. So remember in the first, so if RNA polymerase stopped here, we would have those lovely attenuation loops. Because it is stopped here, we have an anti-attenuation loop forming. Okay, so because this anti-termination loop is formed, RNA polymerase is not blocked. So it can continue on its merry, merry way. Okay, so we've got no tryptophan to bind to the repressor protein, so the repressors can't stop it. We've got no attenuation loop forming, so the attenuation loop can't stop it. So RNA polymerase is finally allowed to continue on to those trip operon genes and transcribe the operon to produce tryptophan. Okay, so I've got another diagram here showing you that. So we've got RNA polymerase starts to transcribe, as per usual. We've got our ribosome just starts translating as per usual. It gets a little bit hairy here because we've got no tryptophan. So that means when we're doing our translating, we do our amino acids and we have to stop. So that means the ribosome is stopped and cannot add to the polypeptide chain anymore. Okay, prematurely stopped here. This doesn't stop RNA polymerase though, this just stops the ribosome. Okay, so that means that RNA polymerase can continue on. This is the formation of this anti-termination structure, anti-attenuation structure, whatever you want to call it. This forms, which does not prevent RNA polymerase from continuing on. Okay, it stops the ribosome, but it does not stop RNA polymerase, which means that it continues on its merry way and transcribes the trip genes, and therefore tryptophan is transcribed and translated and produced. Okay, so if you've got any questions, bring them to class and I will see you there.